Hi, welcome everybody. My name is Rebecca and I work at the University of Leeds, and I head up the biological research facilities within the Faculty of Biological Sciences. And historically, by training, I'm an electron microscopist, and I manage our cryo-EM facility. And I just wanted to welcome all of you here today. So we've got 96 people, 97 still ticking up, of you joining us on the call today for what is, I believe, the 5th webinar in this really exciting series which Instruct have put on over the last couple of months. And for many of you joining us today, you may not necessarily be very familiar with Instruct-ERIC. So I wanted to just kick off with a couple of slides briefly introducing Instruct and what Instruct's about, and some opportunities that might be interesting for you through Instruct. Instruct is a pan-European organisation which facilitates access to structural biology infrastructure, essentially. Instruct is made up of 15 member countries, each of which pay sort of an annual subscription to allow scientists working within those countries to access the full range of services and opportunities that are available through Instruct. And in terms of, I guess, the core offering, Instruct essentially is a fantastic opportunity for researchers to access a whole catalogue of equipment, expertise and resources to facilitate structural molecular biology, whether that's accessing equipment such as electron microscopes, or whether that's accessing sample preparation facilities. There is a huge variety of different techniques available, and through which access is facilitated through Instruct. How do you get access to all of these exciting technologies? Well, if you go to the Instruct website, and the web address is just there below, you'll be able to have a little look at all of the different facilities and technologies which are available. And then in terms of applying for access, it's really simple. You just click on the request access button, fill out a short form, which is then subject to peer review. And then depending on the outcome of that peer review process, you'll then be put in touch with the central technology or site that you've been given access to, and you'll be able to kick off and start accessing those technologies. So really, this is a fantastic way and a fantastic opportunity to gain access to equipment, technologies, expertise that may not necessarily be present in your home institution. Beyond just offering access to technologies or services, Instruct does a whole lot more stuff. So Instruct offers a full catalogue of training activities and hands-on workshops, which are delivered internationally across a huge breadth of research fields. So if you're interested in learning a new skill, in being introduced to a technique, or then there may be some training courses or opportunities that may be suitable for you. So I highly recommend you go and have a look at that. But also Instruct offers, there's a really mixed catalogue. So some of the training opportunities are really focused on expert users, or maybe even infrastructure managers. For example, just a couple of months ago now, we hosted the Instruct Best Practices in Cryo-Electron Microscopy. We hosted that virtually, obviously, because of the present situation. But we were able to bring in expertise and people who are interested in the management of cryo-electron microscopes from all over Europe, but brilliantly from all over the world. We had participants from 3 different continents at that meeting. So this program of training which Instruct provide, there's a whole range and breadth of activities there. So highly recommend you go and have a look. And those courses can offer a great introduction to techniques. Beyond the training catalogue, in some cases, researchers are interested in gaining deep insight into particular techniques and really require access over a sustained period of time, and maybe not just access to equipment, but also expertise. It can sometimes be very difficult to make significant progress on a project within 48 hours. And this is where I think one of the really exciting things Instruct offers is their internship programme. So the Instruct internship programme funds research visits between 3 and 6 months to an Instruct Centre in Europe, and that person can then become embedded at that site and really gain deep training and experience in technique or techniques of their choosing. So that's a fantastic scheme. Highly recommend you go and check out the website if you want more information on that. And finally, Instruct do offer direct sort of direct funding for sort of small scale pilot research projects. And this again is a brilliant scheme that will enable researchers to perhaps have a go at a project and generate some preliminary data that might then be able to prime a larger research grant. So a whole range of different activities and a whole range of different opportunities for you to benefit from Instruct there, and highly recommend you go and check out the website to read more about those. If you want to stay in touch with the latest in terms of opportunities and calls, then if you go and register at Instruct, then you will get all of the latest updates conveniently to your inbox. So as I mentioned at the beginning, I work personally at the University of Leeds and specifically within the Asprey Biostructure laboratory. And although so I had a whole range of research facilities, and we're really excited to offer 3 of these technologies, so mass spectrometry, NMR, and electron microscopy, through Instruct in terms of accepting users and onto our site and to work with you on your projects. And I just wanted to introduce those capabilities very quickly. So in terms of our cryo-electron microscopy access at Leeds, we offer access to our 2 Titan Krios microscopes, and very excitingly, we have new detectors coming online in Q1, Q2 of next year. And of course, we can offer access for experienced electron microscopists to send us their samples, and we can facilitate data acquisition. But I think one of the most exciting opportunities that we offer through Instruct is what we call enhanced support. So this is where you might just have some biochemical analysis of your protein. You might have never done any electron microscopy before ever on that sample. But through this enhanced support pipeline, which we offer through Instruct, you can ship as your sample. In normal times, you could come and visit, and we will be able to guide you through that pipeline, perhaps doing negative stain first and then cryo-EM grid preparation workup. So this is a really fantastic opportunity for researchers who perhaps aren't experts or have any existing knowledge with a particular technology to gain access to these cryo-EM workflows. And secondly, I just wanted to highlight that we also offer mass spectrometry and NMR access as well. Mass spectrometry, there's a whole range of different technologies available, but we particularly offer hydrogen deuterium exchange mass spectrometry analysis through the Instruct catalogue at present. And in terms of our NMR, we have a 950 megahertz magnet and some really interesting and fairly unique capabilities which we make available again through Instruct. So if anybody is interested in electron microscopy, mass spectrometry, NMR, and wants to talk more about their projects, we'd really, really love to hear from you. At the top, there's my own email, r.f.thompson, but you can also email our cryo-EM at Leeds and our research facilities email address to find out more information. So with that, we're going to dive right into the programme today. Without further delay, I'm going to hand you over to our first speaker of the day. So I'd like to introduce, first of all, Dave Stuart, who's going to be talking about the antigenic anatomy of SARS-CoV-2. So, Dave, if I could I possibly invite you to start your camera and then share your...
My camera should be started. I can see myself.
I can see you too, and I can hear you, which is a double plus bonus.
I was a little bit slow in joining because I got 2 invites and I followed the wrong one, which put me as a viewer rather than a participant. So anyway, that was very nice. Thanks, Rebecca. That was a great introduction. Thank you. So I will get going fairly quickly, and I would apologise because as sometimes happens with me, I was putting the slides together in a rush, and which is appropriate because things are changing fairly rapidly at the moment with this field. Okay, yeah, so the antigenic anatomy, which is a funny sort of title, but before I get into that, I'd just like to give a little bit tiny minute of preamble just to sort of really reinforce that the observation that what's become clear during the pandemic is that science infrastructure, including the infrastructure offered by Instruct, has really played a significant role in our response to the pandemic. And the whole globe is relying on science to get them out of the hole. So that's important. But that's particularly relevant to me because I'd just like to give tremendous thanks to everybody that's been involved in Instruct, across many centres, and particularly with respect to my talk, for the people that have contributed to this work, especially in Diamond and Oxford. So I'll be talking about the antigenicity, but I just want to just remind you of something I won't talk about, but Martin Walsh's group and Frank von Delft, together with Neil London and loads of people in Instruct and way beyond, have done a tremendous amount with fragment screening. And I think this is the experimental work was led from Diamond in terms of structure, and a whole bunch of structures were released to the public very early on. And this has been a tremendous example of moving from weak binding fragments to nanomolar leads, genuine leads. So you've gone from fragments to leads in a very short period. So I think there's a lot of work that's been done, and some of these things really have to show as exemplars of what we can do, and we have to try and make sure we don't lose the momentum if we come out of this. But the sort of expertise of infrastructures can help in other ways. So one of the things that Oxford has done is to set up a very high throughput antibody screening facility, which can now screen up to 50,000 tests, do up to 50,000 tests a day. And this was really helped with contacts from the Shearing Institute in Sweden, but also Diamond contributed project management expertise to this project to really help an academic group do something at a sort of semi-industrial scale. So again, there's all sorts of ways that the infrastructures can have an impact. So the body of my talk is going to be dissecting the antibody responses to SARS-CoV-2. And I've not got time to go into the structural results in detail, but just to set the scene, essentially, we were looking at results that came from 42 recovered patients in the Oxford area. And that led to the isolation and characterization of 377 anti-spike antibodies. Of those, we've looked in much greater detail at 80 antibodies that bind to the receptor binding domain, plus another one, which I might just flag. So just to sort of set the scene, I mean, I hope most of you will have sort of have some awareness of the spike now. So the bottom of the picture is where this is attached to the virus. It's a trimeric protein. This is the pre-fusion conformation. So this is the conformation that you want to target with antibodies to neutralize the virus. And the two domains that are of particular interest are the N-terminal domain, which sticks out here on the side of the trimer, and the receptor binding domain, which is on the top. The receptor binding domain, as is in the name, binds the receptor, and there's a well-characterized ACE2 protein on the host cell, which is the receptor. To bind the receptor binding domain has to flip upwards a little bit so that the receptor can then attach to a site that's otherwise rather hidden. So it's a very dynamic molecule. These bits that I've described are part of the S1 chain, because the chain is the single chain is cleaved into two, S1 and S2, by trypsin or by interactions during cell entry. And S2 is the motor that does the fusion. but not involved in receptor binding. So if we cut straight to it, and if we look at the plasma from these recovered patients, we can see that there is, and this is now looking at the binding of the binding of antibodies from these recovered patients to spike, the receptor binding domain of spike, and to nuclear protein, which is a very abundant protein. You can see that as you can, this is diluting out the plasma. Sorry, it should be plasma. You can see that there's strong antibody responses against all of these proteins, and nearly all people mount a reasonable antibody response. Not quite everybody, but nearly everybody. And you can see on the left here that this is now the neutralization against how much you can dilute the neutralization, dilute the plasma and still get neutralization. And you can see that the antibody responses from people that have had severe disease are much stronger than people that just had a mild disease. So over time, you build up strong antibody responses. So this was a fantastic collaboration with a lot of people, including particularly Gavin Screeton's lab in Oxford. And there's a couple of ways that they identified the monoclonal antibodies. Essentially, it's taking single cells, sorting out the single cells that are IgG producing cells, and making sure that those light up, bind to the spike protein, and then just doing standard PCR cloning, transfection, and making the antibody. So this has been done for all of these antibodies. It's 377 antibodies, as I say, and you can see that the slight majority of those antibodies interact with S2, but there's a significant fraction of them, 21%, react with the receptor binding domain. So that's about 80 antibodies from our set. And the other thing to notice, and I'll come back to this, is that normally, if for something like HIV, to get effective immune responses, you have to have a lot of mutations in the variable region to build up tight binding and neutralization. So the number of amino acid substitutions compared to the germline, and that's the original pieces of genetic material that are shuffled together to make the antibody. From that point, you then have somatic mutations that increase the affinity. The number of mutations is actually very low. So many of these antibodies are quite close to germline. And this is sort of good news for vaccine responses, because it means that you can quickly build up. You don't have to do a lot of mutations to build up good antibody responses. And I'll come back to that as well. There is an exception to this. And this is, most of these antibodies are very specific for SARS-CoV-2. There are a very small number, just 20 out of the 377, that are cross-reactive with seasonal coronavirus. That's one of the common cold viruses. And and if you look at the number of mutations there, it's very much different. It's shifted over, and particularly in the heavy chain, there's a much larger number of mutations. So this reflects the fact that there have been affinity matured responses against the seasonal coronavirus. So these are not new antibodies responses. These are things that were against people. This is because people had had a previous cold coronavirus in origin, and some of those antibodies will cross-react with the SARS-CoV-2. So I've said that the large majority of the antibodies are not against the receptor binding domain. So 20% are against the receptor binding domain. But if you look here at the level of neutralization, this is potent neutralization as you go up, no neutralization at the bottom. And you can see that the non-receptor binding domain antibodies, very few of them are potent. There's one that stands out here, which I'll come back to in passing, but most of them are not very potent, whereas a good number of the receptor binding domain antibodies are potent. And you might think, therefore, that those are going to interfere with receptor binding, and that's the mechanism neutralization. And there's some evidence from that. If you look at the level of neutralization here, very potent at this end, poor neutralization here, and then the affinity for the ability to block ACE2 binding, you'll see that the potent neutralizers tend to be good ACE2 blockers. The ones that don't block ACE2 are less good blockers. And in fact, the strength of association between binding the receptor binding domain and being effective at neutralization is shown very strongly here. And this, just look at the orange bars here. So this is now when it says plus RBD. So what's happened here is you take your plasma with all the antibodies in, and you go in with beads which have receptor binding domain attached to them. You shake it around, you take the beads out, and that extracts the antibodies that bind to the receptor binding domain. And you can see that this is for different patients. You can see in this patient, about 90% of the neutralization was lost when you take out receptor binding domain. So the neutralization is pretty heavily focused on that bit of the molecule. And this is the sort of anatomy of that bit of the molecule. This is the fold of the receptor binding domain. And by analogy with this sort of classical torso, we've named bits of it: neck, shoulder, flank. And this is to help us describe where the antibodies bind. And we'll see that the really interesting region is up here, up at the top of the molecule. So having got these 80 receptor binding domain antibodies, we then made the receptor binding domain, attached it to a octet tip. So this is we're now using biolayer interferometry. So what we do is we attach the receptor binding domain to a tip, and then we go in with one antibody, and we see binding. We can read that out in the octet machine. And then we go in with a second antibody to see if that second antibody can still bind in the presence of the first antibody. So that's a competition experiment. And this is a fantastic heroic effort by Darming in the lab, who used a 96-well plate octet machine to do a matrix of comparison between the antibodies. The matrix wasn't quite complete, but he did thousands of experiments. And then Helen Ginn, who was a student with me, is now a Diamond postdoc, very recently published a paper in ACTA on a clustering algorithm for clustering essentially stuff that comes out of the XChem screening program. She applied that clustering algorithm to the competition results and reordered according to cluster. So here, red means strong competition. And you can see that there are several blocks here of antibodies. There's one block, a little block up at the top. This block here, where there's extremely strong cross competition between this block and this block, as seen by the off-diagonal terms. This block cross-reacts with this block, and then this is almost independent. So this is the output of the clustering algorithm. You can see that then there's sort of, this row that cross competes form this sort of arc here in this sort of hyperspace of the clustering algorithm. And then this left flank is separate. So these now, I've mapped these to the torso. How do I map them? Let's let's just describe how we can map them. And this was something that Helen Ginn has again implemented. It struck us that this competition data is very redundant, very information rich. So what we did was Helen made a mesh around the receptor binding domain. We took 3 antibodies I think 4 antibodies of known structure. So some that we determined, and some other people had determined, and added those to the competition mix to give us anchor points, put those down, and then used a Monte Carlo and steepest descent algorithm to find where all of the antibodies bound. And that allowed us to place all of these 80 antibodies onto this receptor binding domain. So this is now the receptor binding domain. And this is just to show where ACE2 in purple binds. The green bit is the occluded surface on the receptor binding domain. So that's the sort of the action bit of the molecule. This now shows the results of Helen's algorithm with Darming's data, and the clusters now are different colors. So the flanks are down here, the blue and the purple. The neck is up here, and then the shoulders, you can see that there. So we've got a complete mapping now of where all of these bind. We can now look and see how the neutralization maps on to these, and blue is not neutralizing through white up to red for the most highly potent neutralizers. And you can see that the potent neutralizers tend to be in the region where binding would block receptor binding. There's some interesting exceptions. Some molecules bind but don't neutralize, and we don't understand the reason for that, but there's a pretty good correlation. So we then did structural analysis, and this is both crystallography at Diamond, and Darming and Wren were the drivers of the crystallography, and also cryo-EM on the whole spike. I haven't got time to show really hardly any of the results, but this is the results abstracted. So in fact, we got a similar number of results from both cryo-EM and crystallography, a total of 19 structures. And this shows where these antibodies bind. And you can immediately see that the majority of them bind, because we focused on the potent neutralizers, bind in such a way that they will block the ACE2 binding as expected. Now, one thing to remember is this is 150 and 158, and they're binding essentially in almost an identical epitope here. And this is a sort of interesting epitope, which I'll say about. But also this one over here, 384, is the most potent of any of the antibodies. And it's got a slightly different angle of attack, different binding position to anything that had been seen before. So that's an interesting antibody that we think may have some therapeutic potential. Now, if we just look at the use of the different V regions of the antibody, this is very complicated, but there's lots of different colors. This shows that there's a pretty broad selection of lots of different bits from the sort of jigsaw, or the collection of possible genes. But what's interesting is if you look here, this brown region, which is 353, is a very commonly used public V region in the antibody. And you can see it's not present again in just the non-RBD binders, but it's comes up very strongly here in the RBD binders in the heavy chain. So this is a particular heavy chain that everybody has. It's very common. Well, maybe not everybody, but it's very common. And it's used to select binders to the receptor binding domain. And now if you look in this region here, red means that it's really potent. So if you look at the antibodies that use this V region, the 353 V region, they're fantastic neutralizers. There's a few others, V regions, that also can be effective neutralizers, and I haven't got time to talk about those, but this is really outstanding. And if you look at the details of this, I won't go through the details. The key thing here is, and this public usage was picked up by Ian Wilson a few months ago, and we find, as he observed, that that because this V region is so dominant, it's the H1 and H2 CDR3s that do the majority of the binding. The CDR3 is actually quite short. It's almost tucked out of the way. That means that despite the fact that there's a lot of, there can be variation in the CDR3s, because it's not dominating the binding, it's the others dominating that don't change so much. The mode of binding of all of these is essentially identical. They bind in the same way. And you can see this if you look at all of the structures that are in the PDB. Nearly all, the vast majority of them, this great cloud here, are these binders that go for that epitope. So they're really overrepresented. So that's the sort of key bit of the story that I wanted to bring out. This is the exception. This is the receptor binding domain is in the middle. This is the N-terminal domain. This is our 159 antibody that is a very strong neutralizer, and yet it binds the N-terminal domain. It's a strong neutralizer, but only neutralizes when the full antibody is present. So the FAB has essentially no neutralizing capacity. Here we've modeled the full antibody on, and you'll see that the two arms here cannot reach across between the N-terminal domains. So our assumption, and we've got no evidence for it, is that the neutralization is by the sort of aggregation or the cross-linking of viruses to produce neutralization. But we don't know. There may be other roles that are not yet understood. So there's lots of interesting stuff that's come out of this work, which I haven't got time to talk about. We find that 17% of the variable regions are glycosylated, and actually removing the sugar reduces the neutralization potency of those antibodies. And we've got structures of those, and we can see that there is interaction between the sugar and the receptor binding domain. But just to sort of finish, this is work from Mike Diamond's lab using a transgenic mouse model, and the simple take-home message here is that if you compare with control, if you treat prophylactically with any of these potent neutralizing antibodies that we found, they protect in this model extremely well. And if you go in a day after infection, so now you're not doing it prophylactically, but following infection, you're seeing if you can treat an existing infection, the most potent antibodies are capable of abolishing the infection in the mouse model. So, and there's a good correlation between the strength of binding, strength of neutralization, and the effectiveness in the mouse model. And just to finish, because for people that have been following the news, I don't know about in Europe, but in the UK, yesterday, the government announced a new variant seems to have come about, and it looks like it's really taking over in some of the areas in the UK, where there is the highest rate of growth in the number of cases. There is one change in the receptor binding domain in that mutant, and that is at residue 501. You can see it is in the ACE2 binding site, and there it is. It's not that, you would think it's not that close to the neutralizing antibody binding sites. So you would think this may be okay. And you'd think also, because it's very close to the ACE2 here, the surface is the ACE2, this is the mutant in the, sorry, the surface is the, surface is the ACE2, this is the mutation in the receptor binding domain. So you'd think it might throw off the ACE2, weaken the ACE2 binding. We don't know. All of this now has to be done to understand it. But this is what is really interesting. I don't want, and you know, this is now, these are those very important public antibodies. This is how they bind. The key thing in the binding I talked about was the heavy chain, but the light chain also interacts. And if you look where the light chain interacts, the light chain in both of these examples that we have structures for, the light chain interacts very strongly, and you can see there are really clear hydrogen bond and hydrophobic interactions with this residue. So it is conceivable that this mutation to a tyrosine from an asparagine could affect the binding of these important antibodies, and that could have some impact on neutralization responses. So I'll finish. I'm sure I've gone on too long. There's a lot of people who've worked on this. Darming, Helen D has done the EM. Helen G has done the mapping. Ren has done a huge amount. Especially the crystallography, but all sorts of things. Liz has been working on pulling all of these things together. Tom has done biophysical characterization. Nui and a whole host of people in Gavin's lab. Mike Diamond and James Brett in his lab have done the animal experiments. And as I said, the people in Diamond have been fantastic in allowing things to carry on through the pandemic. And I will stop. Thanks.
That's great. Thanks very much, David, for a beautiful talk. Lots of really interesting stuff in there. And just want to say, I think it was a great point that you kicked off by, really highlighting how technology and research infrastructure has underpinned the very rapid progress that has been made. I think that's a very poignant point. So we've had a few questions in the chat, so we'll just run through those. So just to kick us off, we've got a question. Are there any population differences in terms of the germline repertoire in different groups of populations that might make particular populations more or less sensitive to SARS-CoV-2?
Well, to be honest, we haven't looked at that properly. There's I didn't have time to talk about it, but it's interesting. There are several important several of these important public V regions, and I think that it'd be very interesting to look at what the distribution of those is in populations and across the world. But we haven't done that.
There's been, well, somebody asked a question around glycosylation, and I thought there was some really interesting, and the question was asked before you came back to the interesting point around the glycosylation. So the question was, can you correlate non-glycosylated regions to better neutralizing antibodies, or does the heavy glycosylation play a role in immune escape? I guess some of the results that you presented suggest that actually glycosylation might be essential in terms of
it's really complicated, because what we found is that the antibodies can be glycosylated, and the glycosylation of antibodies can actually be important, which is something I was sort of blissfully unaware of, really. But the glycosylation on the receptor binding domain, most of these interactions are protein-protein, so they're not using the sugar. There is a sugar on the receptor binding domain that one very potent antibody, S309, uses and is critical. But I'm not aware of, from all the sequencing data that's been done. At one stage we thought that there was a mutation in that sequon that might be associated with escape from, but I think that was largely a sequencing error. So I'm not sure, but yeah, clearly the glycosylation will have an effect, but despite the fact that there is a lot of glycosylation, you saw that the slight majority of antibodies are actually against S2 rather than the receptor binding domain. We could discuss that a lot. Sorry, I'll stop here.
Yeah, well, I think it's really fascinating. Now, are there any efforts underway to really catalogue the glycosylation which is found in more detail? Because I guess this is a little bit of more of an unknown territory.
Well, Max Crispin, who was in Oxford and collaborates with us and with Ian Wilson, is done and is doing some lovely work on this. Yeah, there's a lot of work going on.
So, more technical question, which I guess has implications for the throughput of the structural biology. So, someone has asked, in terms of the crystallization screening, are you seeing a wide variety in terms of the conditions which are coming out of those screens, or are you finding that actually similar conditions are enabling the crystallization of different antibodies?
I'd have to look in detail at the crystallization section. Generally similar, but it's the and there's similarities in the crystal forms, but they're not all identical. And in some cases, and it's the crystallization has not been terribly straightforward. In some cases, we have to Ren and Darming have made binary complexes. In several cases, and again, I didn't have chance to show them. In several cases, the crystal was a ternary complex of two Fabs plus the receptor binding domain. And that's one of the interesting things that from this competition data, you can also predict which Fabs can bind at the same time. So it would help in making cocktails for therapy. Because again, having separate binding sites is going to be important to avoid escape.
No, it's really interesting. And I guess just as a final question, apologies, we're not going to be able to get through all of the questions in the chat. I guess the speed at which this research field has progressed over the last months has been unprecedented. I guess, do you have any final comments on, what you think the next stages are going to be for this field, what the priorities are, and specifically, I guess, what role Instruct might play in that going forwards?
I think you should move on to the next talk because I had a long time discussing that. I think what's important is that we try and learn lessons from this and work out where we've been strong, where the gaps are, and what we need to do to sort of be a bit more robust about this. But I think it's been a great, as I said at the beginning, and as you said, it's been a great selling point really for these for infrastructures and for structural biology and molecular techniques as a whole. So we shouldn't just sort of move on at the end of this. We should reflect and try and be ready for the next disaster. Even if it doesn't come, we should be ready.
No, that's great. Thank you very much. It's a brilliant talk. Great. And now we're going to hand over to Ray, who's going to talk to us in a little bit more detail about the neutralisation by nanobodies.
Okay, thanks very much, Becky, and Instruct for the opportunity to give an update on what we've been doing with the nanobody technology platform. My first slide, I've just some images of the buildings that we've been working in, just really to acknowledge that this has been a great team effort between Dave's lab in Struwe, my group and Jim Naismith in the Research Complex, and obviously collaborators at Diamond, where we're based at the Research Complex. And the final image at the right is the new Rosalind Franklin Institute, which I just thought I'd introduce, which is now basically the home of the nanobody discovery platform, which I'm going to talk about. I had a few introductory comments, really echoing what's already been said. SARS obviously had a major impact, and although it's been close related to other viruses in the same area, there's something different about SARS, and I think we haven't really unpicked that completely, although the fantastic progress on the immunoresponse as Dave's just been outlining. And I think the other general point to make is there's been a fantastic, unprecedented, in my opinion, sharing of unpublished data, which has allowed the scientific community to respond very quickly to the pandemic. And as Dave also said, I think the lessons to learn about how we've done that, and obviously lots of positives as well. And I think this information has allowed us to get in quickly, particular strategies of producing the molecules we're working with, to look at the structure and obviously lead to the ultimate goal to antiviral inhibitors. And really the motivation to work on this in the nanobody field is really to drive the production of potential therapeutics and diagnostics. Probably don't need to remind this audience about nanobody technology. It's really a core platform within the Instruct offering. But just to say that they're unique agents, they derive from the heavy chain only antibodies of camelids, llamas, alpacas, and camels, which is like in llamas about 10 to 15% of these rather curious heavy chain only antibodies. And the nanobody bit is derived from the variable domain of that heavy chain only antibody as shown in the cartoon at the bottom. The big advantage, and this all goes back to work that was done, pioneering work on discovery of this somewhat serendipitously in the group in Brussels, and has been taken forward hugely in the structural biology space by Jan Stuart and his group at VUB. The nanobodies have big advantages because they're small, single domain binders, and they've found many uses now in biomedical research, from microscopy through imaging, but particularly in structural biology, where their ability to bind and stabilize conformational shapes of different molecules, particularly membrane proteins, I would say has been transformative. And again, this is the work from Jan Stuart's team. So just to give you a walk through the nanobody discovery workflow that we've set up at Oxford and Harwell over the last 18 months or so. There are two ways that we're making nanobodies. One, the route A, shown here, is the conventional through immunization. I'll come back to that at the end of the talk. This obviously has an elapsed time because you've got to go through a certain degree of prime boosting. But the bottom line, what you take from that is to derive a library of the VHHs, just those single domains, present them in some display strategy. We're using phage display as a standard route, and then screen by panning against a particular antigen or antigen complex. The other way of doing it is to actually start not with an immunized library, but with a library from a non-immunized or so-called naive animal, or a bunch of naive animals. We've got several of these libraries now, and just interrogate that with your antigen in a fishing exercise and see what can be pulled out. And then we've shown, and many other people, that there are weak binders that can be isolated this way, but you do then need some way of improving their affinity. And so we have a process of affinity maturation based on PCR mutagenesis of the sequences in the CDR3. A lot of work on structures and function, showing that the CDR3, the third complementary defining region of the nanobody, is the main interactor with the antigens. That's where most of the action goes. So we've chosen just to mutate that particular sequence with a sliding window, really changing every residue in the CDR of our parent molecule. And then you go through a process of screening, sequencing to bin, because you're looking for convergence around particular sequences, then ultimately characterization through, in our case, SPR or BLI, as Dave mentioned, and structural studies beyond that point. Another key thing, apart from having the kind of workflows to generate these reagents, is obviously having the antigens to put into the system. I just thought, as a protein production person, just to name check on this, the two reagents we've been working with primarily are the receptor binding domain, which you've heard about, and the full spike protein. Going back to March, and I think Dave would agree that I think there's a lot of work went in, both in my lab and Struwe, to work out how to make good quality protein. And I think this is essential for obviously the structural work, but also for the functional studies and investigating antibody-antigen interactions. So we use the suspension HEP-XP cell system, get really good deals of these materials transiently. And for the spike protein, apart from EM, we use SecMals to characterize it, because obviously looking to form an authenticated trimer, which is obviously stabilized by these protein mutations within the sequence. So these are two kind of antigens in the system for both screening and analysis. So what you get from your naive screen is a lot of potential hits. We screen with a phage ELISA, so we're using the phage presentation of the VHH as the reagent to immobilize the captured biotin captured antigen on a plate. This just shows you the sort of spectrum of absorbance in a simple assay. And I just want to, we put an arbitrary line here at OD2 to distinguish things that are probably very weak to things that might be a bit stronger binders. There's a lot of hits here, so we're just going to focus on H11 and I'll tell you a bit more about the H11 series that we developed from this parent molecule. So in the right-hand table, there's a series of affinity matured variants of H11, in which, as you can see from the KD, we've gone from around 400 nanomolar binding KD determined by SPR for the parent, right down to H11H4, where we pushed it by nearly 2 orders of magnitude to a KD of now 5 nanomolar. And that's all by mutagenesis in vitro. So these are the molecules we're going to talk about in a little bit more detail, particularly focusing on the most potent ones where we've done the most work. And this was published earlier this year. So one of the first things we investigated was whether any of these molecules interfered with the binding between the receptor binding domain and AC2. And obviously it's been emphasized in Dave's talk how important neutralizing responses in man have been to actually interacting directly between AC2 and RBD. This sensogram just shows you that these antibodies, or one of these antibodies, which characterized all of them now, actually block the interaction between RBD in solution to immobilize AC2 on a sensor chip. So we make AC2 as an FC fusion shown in the SDS gel on the left of the panel here. So we have a binder which interferes with AC2 binding to RBD. And this has been shown in a different way. This is in collaboration with Anne Townsend's group at the WIM in Oxford, at the university. This actually basically just turns the thing the other way around, where actually the RBD made as a multimer using spy catcher technology on a VLP is actual antigen, and we're looking to compete out soluble AC2FC, which is labeled with streptavidin. And on the graph here, you can see as the slope, as you increase the concentration, but actually in this case from right to left, you get an inhibition of the interaction between soluble AC2FC and this multimer, which is a sort of mimic, if you will, of the viral surface, because it expresses multiple copies of the RBD construct. The other thing we looked at, and this is going back before all the data that's now emerged about where these different human antibodies bind, the only reference material that was available way back in February was an antibody called CR3022, which is a cross-reactive. It was raised by a phage display on a single chain FV from a SARS-1 patient. And essentially this was shown by others that it cross-reacts with SARS-2, and there's structural work being done by Ian Wilson's group and Dave's as well. So we use this as a reference because it binds, it's known to bind not to the RBD, I can't remember on Dave's anatomy, but it binds the distal to that. It's right down at the bottom end, if you will, of the RBD domain. And here we showed that our molecule did not compete with CR32 binding. So it placed it not at that part of the molecule, but more likely at the AC2 binding site at the top, or the shoulder part, as in the molecule model. And we could show this directly, obviously, by structural work. And so on the left-hand side is just an overlay of the H4D4 nanobody in gold bound to the RBD, which is the sort of green, and then superimposing that on the structure that's already been published on RBD interaction with the actual AC2 itself. And you can see that we're binding close to the interaction surface between RBD and AC2, but not directly onto that surface. It's actually somewhat to the side. And it appears that it basically gets in the way of AC2 getting access. That's our sort of working model of how this is actually interacting. I should say that all the interaction sites that we've mapped now are different from the ones that have been drifting through mutagenesis. So at the moment, we have no evidence, at least, that some of the natural mutants that are appearing would interfere with interaction in this case. And on the left side is the co-structure of the CR322 RBD, and in this case H11H4 nanobody, just showing very directly the different positions on the RBD that these two molecules bind to, so completely opposite ends of the RBD. And working in collaboration with EBIC, Diamond, and Dave's group, and work in Jim's group, we've been studying the interaction between this and many nanobodies with the spike protein. This just is showing a EM structure of the spike protein bound to the H11H4 nanobodies. The modern bodies are obviously not well resolved, so that's the electron density has been put in from the X-ray structures, just showing the combination of EM and X-ray crystallography in developing these models. And essentially what we see here is the typical 1 up, 2 down conformation of the spike protein, the RBDs in different appositions. and the binding of the H11 nanobody to each either up or down RBD. So what's emerging from a number of structures is that the nanobody at least can access the RBD in either the up or down conformation. And in actual fact, the particles which were imaged for this model essentially were all in one conformation. In this case, it was 2 down, 1 up. So it also seems that interaction with nanobodies, and it may well be the same case with antibodies or FABs, they do lock the spike protein in a specific conformation, which otherwise would be quite dynamic. The other thing I can say now is that we've modeled in an all down RBD version of the spike, and you can actually get access to the nanobodies to that sequence. And really in more recent work with other nanobodies, we've shown that we can show binding to an all down conformation. So mechanistically, we think how these nanobodies may work against the whole spike is by locking it into this unproductive, non-binding state. And the proof, if you like, is really what happens when you expose these reagents in an assay to investigate virus neutralization in vitro. Just to explain what this assay involves, it's quite old school in the sense that basically you're infecting vero cells which express AC2 with live virus, looking for plaque formation. The cells are grown in a semi-solid support and they're stained in the 24-well plate. On the right of the image here just shows you the different plaques, and you're looking for clearance and plaque number reduction. So plaques are counted manually in the case of this assay, and then plotted against the concentration. So based on the concentration dilution series shown here, this is work done in collaboration with colleagues at Public Health England at Porton Down. The IC50 for this H11 molecule is around 6 nanomolar. Now, this is expressed not as a single antibody, but as a nanobody fused to a human FC, which is quite a traditional way of increasing the avidity of nanobodies and also recreating effectively a whole heavy chain only antibody species. Now, at the time, this is going back now a few months, this was one of the most potent neutralizing activities that have been disclosed at the time. I think a lot of the human antibodies now have come through with increased potency, but at the time, this was, I say, a bit of a pathfinder and also obviously based on a nanobody species only. Given that we've had this affinity maturation series, we've sort of developed a side project really to understand a little bit, if we can, how affinity maturation by mutagenesis has driven the affinity improvements that we've seen between the H11 parent and the series that I've been talking about all the way down to H11, H4. And some very elegant work's been done on using ITC to investigate the thermodynamics of this interaction. I haven't got time to go into great detail, but if you look at the delta H and delta T, delta S, the enthalpy and entropy contributions to that interaction, you can see that's quite variable. The H11, the enthalpy is quite low compared to the other series, so that may account for some of the weaker interactions. But there's clearly an offset here between entropy and enthalpy and the penalties therewith. And what it seems, if we look at the last line, H11, H4, we've got a bit of a compromise here where there's good enthalpy, but not a big penalty compared to the others in terms of entropy loss. So there is some subtlety here in the interactions, but it just can be explained in thermodynamic terms. The other thing to say is that these are relatively stable molecules, as we know from nanobodies. TMs are around 60 degrees or more. There are still the structures of these different complexes, and on the bottom here, actually the ITC, the thermodynamic calculations for KDs, which are similar but not exactly the same as the kinetic KDs. The pattern is the same. We're going from around a micromolar, a half to 1 micromolar, down to a few nanomolar binders. As you'd expect, the structures are pretty similar, and what we see, without going into great detail, is that the positioning of the CDR3, which is the main interacting sequence, is not changed significantly at all within these different complexes. So the actual reason for the affinity gain, apart from thermodynamics, is much more subtle. There are some changes with slight shifts in the position between the nanobody and the RBD, which again will probably contribute and be part of the explanation for the change in binding that we see. So try to investigate this in a little more detail. Essentially, this just shows you where the points of interaction are. And again, with the interest of time, I won't go into details on this slide, but there's a key salt bridge in CDR2. So there's a contribution from CDR2, but the main interactions shown in the dark colors and reds, the hydrogen bonding networks that are established between the target and the CDR3 loop itself. And there's a gain of one significant hydrogen bonding network within H4 compared to the parent, which may go some way to explain this uplift in affinity. So in the last couple of slides, I just wanted to introduce what we're doing now, what's going forward from this study. As I mentioned at the beginning, the two routes to nanobodies, there's a sort of in vitro route, which I've described, but also immunization being the more traditional route. We set out to do both in parallel. Obviously, with the in vitro route, we could get there much more quickly, and it's really given us the information, insights of how nanobodies might be used to investigate the structure and function of the SARS-CoV-2. Anyway, we have a second generation of nanobodies being developed through immunization, through immunizing with the RBD spike combination. Here we're seeing different epitopes, or more epitopes, as you might imagine, from a natural immune response. And the affinity of these agents is significantly better than the first generation VHHs. Again, as you'd expect, obviously the affinity maturation response in the animal. I'll just give you a snapshot of some data that we've generated in the table. These are 4 of the reagents I've mentioned. And what we did here was to try and bin them according to their interaction or competition against either AC2, the sort of first generation H4, our best nanobody of the time, and CR322 as our reference for, if you like, the opposite binding surface to the AC2 interaction site. And as you can see, we're seeing different patterns here. We've got antibodies that are clearly interaction at the CR322 epitope region, and antibodies that are interacting at the AC2, and some of which are AC2 blocking, but not actually directly competitive with AC2. So some interesting molecules emerging here from the llama. If you like, it's somewhat parallel to what we heard from Dave, that's emerging from the natural human response. This sensorgram just gives you an example of the data. I think what's particularly exciting for us now is we've taken some of these reagents into viral neutralization experiments, and I'll just give you a headline result here. Again, reconstructing a nanobody FC as a divalent high avidity reagent. The IC50 now in the plaque reduction assay I mentioned earlier is now down to 2 picomolar. So the IC50 is 2 picomolar. So there was this several thousand improvement in our first generation nanomolar binders. I think the other striking thing from these data is if you look at the curve, it plateaus out at 100% neutralization. So these reagents are actually able to completely stop the virus in its tracks and block all infectivity. And the plate on the left just makes this point very dramatically. There are no plaques seen in exposure of the virus and cells to these particular reagents. So in summary, I would say combining the naive screening with PCR mutagenesis, we were able to very rapidly, and I'm talking in a matter of weeks, to generate nanobodies that block receptor binding and were useful probes to understand that interaction. And it was particularly focused on the receptor binding site. And as is seen now with lots of antibodies, interacting at that site does bring neutralization of the virus in vitro assays. The single particle EM and crystallography, etc., has contributed to our understanding of how these nanobodies interact and what might be underlying their mechanism of potent activity. But I think more strikingly and more importantly is the second generation of reagents that we're now developing through immunization, which are very potent neutralizers of the virus and have great potential, and already that potential is being realized as candidates that could go forward into the therapeutic and diagnostic evaluation. And I should say, I think there's a room for whole antibodies and antibody fragments and different reagents. I think the big value that we see with nanobodies, and it's been emphasized by others, is their size. They are easily produced in microbial systems, so the cost of goods is much less than whole antibodies. And their ability to penetrate tissues and cross the mucosa means that topical delivery through the airways as a nebulizer is a very attractive and realistic prospect of an interactor like this that can neutralize a virus in vivo. A lot of people involved in this study. I'd particularly like to highlight Yidang Hu, who's really basically been the nanobody driver. He's really contributed a lot and produced these reagents. Big team at Harwell involved in making reagents, doing all the structural work at Porton Down and Oxford and Dave's group and the WIM. And thanks very much for listening.
That's great. Thank you so much, Ray. That was a really, really interesting talk. Lots of interesting data in there. So we just had a couple of questions through in the chat. So the first one is relating to essentially receptor binding. So I guess more broadly, the question is, do you think we've elucidated everything that we need to around what are the which receptors actually play key roles in SARS-CoV-2 infection, and somebody's particularly asked around nicotinic receptors and whether they have a role to play.
I think I can only comment on what we've seen, that if you can stop the interaction between the virus and AC2 on a cell surface, then you'll stop infection. I think the big concern is whether the virus will get around that problem, and I think it depends where you're binding at that interface. And clearly, if you can bind at a point where the virus cannot get away from its ability, it can't mutate away from the AC2 interaction, then I think we're probably on a reasonable good track. But the virus may find other ways of doing this, which we don't know yet, of course.
So that really nicely leads us on to the next question, which is specifically around the H11H4, which appears to bind on the side. So do you think that because of its binding orientation, there is the potential for it to also interact directly with ACE2?
We haven't seen that. We haven't looked at that. We haven't seen that for sure. I should say we generally are experimentally looking at whether the virus can get away from these types of antibodies for sure. And so I think, and we're also looking at the interaction with, cross-reactivity and the mutants that have been characterized, like the 501 that was mentioned earlier. At the moment, we haven't seen any escape, at least in the kind of taking a known mutant and trying to see if it will bind still, but work in progress.
So just to finish off, I guess it's really exciting that you've been able to identify these nanobodies with such high neutralizing potential. What you mentioned, obviously, the potential for these to be developed into therapeutics. What do you see as the next stages for this work?
This is kind of work in progress. I can't say too much at the moment, but basically what you would have to do is to take them into the animal model. There's a very well-established now model in the Syrian hamster for looking at actual infection with SARS-CoV-2 and looking at pathologies and stuff like that. So there's an obvious route to go from here, and I think that's the next step in this type of study.
Without further ado, I'm going to hand over to Weston as our next speaker. Thank you.
Okay. Thanks, Becky. Thanks, Pauline and the organizers. I'm going to talk about two projects today, two COVID-related projects, and I will cover in a little bit of some of our previous research results in HIV, because it's no doubt relevant to the work that we're currently doing. So before I get into the heart of the research, I'm going to just highlight the two methodologies that we use, mass spectrometry, which I'm sure, of course, everyone's familiar with, and mass photometry. So we use two different types of mass measurement techniques to look at the assembly, interaction, and heterogeneity of different proteins and protein assemblies. And I think because mass photometry will probably be new to most, I'm just going to take a few minutes and highlight what mass photometry is. So in the simplest definition, mass photometry is the measuring of mass with light. So specifically, the way that we do this is we focus light on a sample volume. So this is protein samples or biomolecules in solution that are on top of the glass cover slip. Most of this incident light travels through the sample. What we're measuring is back reflected light as well as scattered light. And the scattered light occurs when a protein interacts or lands on a glass cover slip, and this results in scattered light. So we measure the interference of both the scattered and the reflected light, and what we get is differential contrast. This is what the differential contrast looks like. So this is the image. This is what the sample sort of delivery system looks like. So the sample is on top of the glass cover slip, and as we take this image, which this is the raw data, we see the proteins landing on the glass cover slip, and if we plot these, the measured contrast as a function of time, we can convert this to a mass distribution. So shown here is counts versus mass. So we can see and measure the relative amounts and mass of proteins and protein complexes. What we've shown earlier this year is the ability to directly measure binding affinities. And this is an example of an IgG1 binding to the high affinity FcR1 receptor. And you can see not only differences in complexes based on the pH, but we can use these data to calculate dissociation constants, of each of these individual binding events. So you can see the IgG-Fc domain binding 1 or 2 receptors. So coming back to our project, we had some very simple questions that we were interested in answering. Specifically, what is the interaction at the host cell interface? Namely, how is the viral spike interacting with ACE2? What are the stoichiometries of these interactions? Is the spike binding a single ACE2 receptor? Is it binding multiple ACE2 receptors? Does it preferentially bind an ACE2 dimer over an ACE2 monomer? What is the role of the ACE2 monomer dimer in equilibrium? We've also been looking at the physiological relevance of the ACE2 monomer dimer through in vivo, as well as infectivity assays, which I'm not going to present today because this is ongoing research. But just to highlight some of the aspects that Dave and Ray have discussed, the spike exists in this pre-fusion down and up state where the RBDs can be accessible to ACE2 when they're in some configuration. And this, of course, can affect how ACE2 binds depending on the number of RBDs that are up, so to speak. We were also motivated by a previous research looking at HIV by mass spectrometry, specifically how an antiviral lectin binds and neutralizes HIV. So we were able to track the number of bandlets, the viral that we were studying, the number of bandlets bound to a single end spike, shown here in orange, but also the observation in the occurrence of cross-linking due to bandlet binding to multiple ends. And from these data, we were able to quantify the binding affinities and observe some degree of cooperativity. And I think what's important to note is when we tried to do this structurally with cryo-EM, all we saw was a pellet. So we were effectively able to measure something where we were previously seeing aggregation. We were also motivated by some of the nice high-resolution structures that were coming out. Here's an example of the viral spike with one RBD in the out position and the ACE2 dimer. But we were also interested in looking at the various models that have been proposed from these structures, whether it's an ACE2 dimer binding two different spikes separately, or a single spike that was interacting with two different ACE2 dimers, or the possibility for multiple interactions and some degree of cross-linking, whether it's through different RBDs on adjacent spikes, or simple interaction between one ACE2 dimer and a single spike. We have increased avidity through each ACE2 monomer binding a single RBD on an individual spike. So the first thing that we did was we looked at these two. So this is the soluble domain that contains the peptidase domain as well as the NEC domain. So this is in the absence of the transmembrane cytoplasmic neuron. And you can see quite clearly the existence of a monomer dimer ACE2 at the concentrations that we're measuring, which is at the low nanomolar range. And we could calculate Kb approximately per nanomolar. So we can see quite clearly this is existing in two different oligomeric states. In the absence of the NEC domain, this is purely monomeric. And we wanted to make sure that these were, in fact, active proteins. So we looked at binding to the RBD and to the rest of the geometry, and we can calculate these KBs quite easily as we've done in the past. And we can see that the binding strengths are low nanomolar, which are consistent with SPR measurements that have been published. The only difference is we can see and quantify the individual binding events. So whether it's the first RBD binding event or the second RBD binding event, and we didn't see really any significant difference between these interactions. When we looked at the spike, we saw, and this is the soluble domain of the stabilized spike that we've been discussing and seen in Dave's talk. We saw higher order oligomers. So not only just a single trimer, but a dimer, trimer, tetramer, pentamer of trimers. And we were somewhat happy to see this publication from Andrew Ward's scripts that looked at oligomerization. So he also observed dimer and trimers, and could explain how these oligomers are forming. And I think what was secondly important about this publication is they observed that all of the RBDs were in the down configuration from their structures, but these proteins were binding ACE2 from either ELISA or biointerferometry measurements. So they're clearly active and implies that the RBD is in fact highly dynamic, moving from an open and down configuration, open or down configuration quite readily. So what we did then next is we looked at monomeric ACE2, and we could see that we had up to 3 equivalent bound, suggesting that we could see this 3 RBDs in that position, a number of binding to the dimer of trimer, but we weren't really able to resolve anything beyond that. It's because these peaks become quite broad once they start binding different numbers of monomeric ACE2. With the wild type ACE2, so to speak, we could see what would appear to be an ACE2 dimer binding, and I should say that at this point we don't know if this is an ACE2 dimer or 2 ACE2 monomers bound. We don't have that structural information, but I will highlight that we only see even number of ACE2 bound. So it appears to us, and this is something that we're following up, that the spike does preferentially bind ACE2 dimer, at least in this experiment. If we look at the raw data, we can kind of get a very easy visualization of what may be happening. So if we look at the spike, you can see each of the waning events. And if you look closely, you see that some of the contrasts are different. Some of the spots are brighter than others. And if we add monomeric ACE2, there's a little bit of, not noise per se, but the ACE2 in the background, and the protein looks relatively happy, and the contrast does appear to be slightly larger if you use imagination. What we start to see, you see these things that we commonly refer to as floaters. Large complexes or aggregates that are starting to form. And when we look at the wild type ACE2, these floaters, aggregates or complexes are much more abundant. And you can see these form. Sometimes they land and interact with the glass surface and stick around. So this is telling us almost immediately that we do in fact have large complexes and cross-linking from the presence of the wild type ACE2. We try to think about how we would interrogate these data a little bit further, and the way that we've started looking at it is through just the number of counts. So you can see on the y-axis here, particularly with the trimer, these counts are around 600. In the presence of the different ACE2s, these numbers clearly drop. So we started to quantify simply just counting the number of counts in these different regions. So either the trimer monomer, from the dimer to the pentamer, shown here, and then anything beyond that. And we see with ACE2 that the number of counts of these higher order oligomers is most certainly higher than in the absence of, and nothing significantly different between monomeric ACE2 and the spike alone. So what this is telling us is that there is in fact cross-linking between ACE2 and spike. And we did this in a little bit more detail looking at titration and plotting these data. And we can see that around a stoichiometry of about 2 to 1 ACE2 to spike, these numbers don't really increase. And we can see the converse of that. At that concentration, we start to see an increase in the number of counts of these complexes. So it seemed to be, and we're still analyzing these data, that at a 2 to 1 ratio, we start to see cross-linking spikes from an ACE2 dimer. Now, I think just in the interest of time, and something that I really want to talk about is, and this has come up earlier in Dave's talk, is glycosylation of the viral spike. So the viral spike has 66 N-glycans per trimer, and I think one of the nice ways that we can view this is from this paper from Rami Amaro and Luis Lufada that showed three-dimensional landscape that's occupied by glycans. So this is an empty model showing us what the glycan shield looks like. And they've also highlighted that glycans are seemingly important. And I think we can also draw comparisons from what we know in HIV and why it's important to look at glycosylation or spike derived from virus opposed to recombinant or vaccine material. And this is something we did a couple of years ago when we looked at glycan analysis, and these colors will be a little bit more evident in a minute or two. But we looked at differences in site-specific glycosylation between the HIV and viral spike from infectious virions, or those from a lead candidate that we produced in hector-chosen. So you can see that the differences are there. And with HIV, this we know is important because many broadly neutralizing antibodies incorporate glycans as part of their binding epitope. And I think as more structural information emerges from SARS-CoV-2, we will perhaps know more about how glycans play a role in neutralizing antibody responses. And we do already know that S309, as David mentioned, does interact with a specific glycan. So I think it's important to consider glycoproteomics as part of our structural biology approach. And there are several examples of this having been done, glycoproteomics and site-specific analyses. But these are mostly of recombinant, these are recombinant material done in a couple different ways. So one example looked at S1 and S2 subunits that were produced and expressed individually. Two different analyses of the soluble domain of the S trimer using a stabilized form. And what that means is the furin cleavage site, which primes the spike for host fusion, has been removed. And these two codons have been added, which sit at the top of the central helix in the S2 domain. So this maintains the viral spike in a pre-fusion state. And what's important is the glycan analysis of these studies are very different. And we don't really know how these compare to those that are present on the virus. And I think before I get into the meat of the results, it's appropriate for me to give a crash course in protein glycosylation so you can understand the data that I'm about to present a little bit better. So N glycosylation occurs in four key steps in eukaryotes. And this is a process that starts in the nucleus reticulum by the formation of this lipid-linked receptor precursor that consists of 9 mannose residues, 3 glucose residues here in blue, these blue circles, 2 unacyl-glucosamine residues. The second step is the transfer of this precursor to a nascent protein in ER, followed thirdly by trimming of these of the mannose residues as well as the glucose residues. And this gives rise to one class of aminoproteins that are termed oligomannose or high-mannose type sugars. And then fourthly, we have further processing or trimming of the mannose residues. We can have what are called hybrid glycans, which are a mixture of the oligomannose type and the complex glycans. The complex glycans are structures that can be largely branched. You can have up to 4 or 5 branches, caprosilic acids, different degrees of glycosylation. And these steps occur in the trans and medial Golgi. It's also important to note that O-glycosylation occurs throughout the Golgi as well. So the way that we present these data is by the 3 different classes, oligomannose, hybrid, and complex, and the different degrees of processing. So green to purple, green being relatively unprocessed or underprocessed, and purple being more complex type glycans. And we can quantify these, the degree of processing relatively easily. So here's an example of HIV-N. We remove the glycans enzymatically with PMJZF, and we label them fluorescently in a 1-to-1 stoichiometry. And we separate these and profile them using hydrophilic interaction chromatography. It looks like this, and this essentially separates the glycans based on their size and shape. If we take an aliquot of that sample, we treat it endo-H, which cleaves with the ketobiased core, effectively removing the ketobiased core of oligomannose type glycans. We essentially remove these types of sugars, leaving us the complex glycans. And we can rerun those. And from integrating the area of these peaks, we can quantify the different degrees of processing. So what we actually did is we took the England strain and we infected cowrie cells, which are human lung epithelial cells, collected the viruses, solubilized the membrane in detergent, purified the spike using the CR3022 antibody, which Ray has mentioned. You can see that there's the S1 and S2 subunits, as well as a tracing like PSA. And then we profiled the glycan for HPLC, and we could see that we had 79% of the triple complex. And the green represent the orthogonal glycans. And this is significantly more processing than we see with HIV, for example. We then did a bottom-up mass spectrometry proteoforms analysis to identify what glycan structures were at what site, and we focused largely on S1 because this is the major immunogen targeted by most of the neutralizing antibodies that Davis talked about. And then secondly, we were also curious to see how the glycosylation of the spike compares using a construct that's essentially the same as the Childhox vaccine. So this has a CMV promoter, the tissue plasma activator leader sequence, the S sequence from the Wuhan strain. This maintains the furin cleavage site and doesn't have this triple chromatin mutation. We expressed this in HEP2 and 3 cells. And what we saw was an abundance of S1 in the supernatant. So that's what this gel is. We looked at what these what the S1 assembly was with mass spectrometry. We see that this is mostly monomeric. This is the cellular domain of the recombinant version. This has a mass of 530 kilodaltons. And the N-glycan processing was significantly more than what we saw in the so we had 96% glycans that were complex, opposed to 4% that were non-homogeneous, but predominantly in this manufactured structure. So on the brink of becoming more processed complex glycans. And then we did the same. We did site-specific glycan analysis to identify what the glycans were, and I'm going to highlight just a couple sites. So in the middle here, we have our site-specific analysis of S1 from the virus. This is our site-specific analysis from the vaccine-derived S1. This is the recombinant trimer that people have been studying, so we expressed it, purified it, and analyzed it in the same way. And the two sites that I want to talk about is the N-link glycan at position 234 and the O-link glycan at position T678. And again, this is just representative for processing. So at N234, we can see that most of the glycans, at least on the stabilized recombinant trimer, are in a MAN9, MAN8, highly unprocessed state. And as we go towards the vaccine, we see this shift towards the MAN5 type glycan. And on the vaccine, this is predominantly MAN5 with a degree of complex glycans. With the O glycan, this is non-occupied in the recombinant trimer. We do see some GalNAc and Core1 O glycans cropping up on the virus. And then it kind of goes haywire on the vaccine derived. So we see not only more O glycans, but an increased diversity of these structures. And the reason that this is important is the position of these glycans. So the N glycan in N234 sits in a pocket that's created by two neighboring RBDs, as well as the internal domain. So this is a MAN5 glycan that's modeled in here. So what we think is happening is this glycan is not being processed on these stable trimers because it's sitting in here and it's not accessible to the ER, the Golgi, and the cytases. So it won't get trimmed when it will be maintained in its organelle state. Whereas with the O glycan, 678, this is adjacent to the furin cleavage site. So what we think is happening here is on the stable trimer, this residue is not accessible to the number of N-acetyl-galactosamine transferases in the Golgi, which is 22. So this is largely driven by protein architecture, sterics, and accessibility of these enzymes to a given protein. So as we look at the vaccine, this site becomes more available, as it were. So to test this hypothesis, we looked at the S1 subunit expressed on its own. And sure enough, the N234 site becomes completely complex. So you can see bi, tri, and tetrametarian glycans. And the O glycan at 678 becomes completely occupied, and the degree of complexity increases. So we see a complete reversal in both the N glycan processing at 234 and O glycan occupancy at 678. So the reason that we think this is important, the way that we can rationalize this, knowing what we know about the biosynthetic pathway, and from this site-specific glycan analysis, if we look at how the virus is derived, the spike is assembled and then glycosylated in the ER. The virus begins to assemble in the ER Golgi intermediate compartment. We have high mannose trimming and cis Golgi, the beginning of complex glycan formation, branching elongation, and the media, and then in the trans Golgi, also methylation. In the trans Golgi is where the virus will encounter furin, and then it'll exit the lysosome. With our antigen, vaccine antigen construct, which has the TPA sequence, the spike is, again, trimerized glycosylated in ER, overexpressed, moving towards the cis medial and trans Golgi, where it's processed in the same way, albeit slightly different. And we rationalize this with a degree of flexibility that exists on the vaccine candidate spike compared to the virus or the recombinant stabilized trimer. Once it's in the trans Golgi, again, it will encounter furin, becomes cleaved, will shed S1, and now that 678 site becomes more accessible to O-glycosylation enzymes in the trans Golgi, and we see this S1 shedding from the cell surface. And there's one thing that I want to highlight beyond just what we've seen from the glycan analysis perspective, but this seemingly makes an important, has an important effect in how the vaccine is derived. So this is a paper, right? Well, it's essentially a Johnson & Johnson vaccine, and they looked at precisely this. So with a spike that's not modified in any way, so this doesn't have the furin. This has the furin site, it doesn't have any stabilizing mutation. It has the wild type leader sequence from the S protein as well as the TPA sequence. You see a modest BNF titer, and this is a non-human primates. Without the wild type signal peptide from the virus, this is essentially the same as the control. With the furin cleavage site removed and the proteins added, you see an increase from there, and then the best response was in the absence of the TPA sequence. And they also did show that the FC effector functions were also best for this construct. So this does obviously have implications for how S is not only assemble the interactions that it may have with the neutralizing antibody, but also for antigen presentation on the vaccine. So just to conclude, with the first part of mass spectrometry project, we can clearly see a monomer-dimer equilibrium of H2, and I think this was interesting because we really struggled to understand the interplay of this protein complex. I will highlight that this is the cellular domain, so one thing I'd be interested to look at is the effect of the transmembrane domain in the lipid bilayer in this assembly. We can see higher order spikes, as well as they're correlated, that's consistent with some Crowley structures, as I highlighted from the reward. It does appear to be that the spike preferentially binds ACE2 dimers, and this is something that we're working to confirm. And then the cross-linking is most certainly occurring, and we think this may be happening in a ratio of 2 to 1 ACE2 dimer to spike. And we're trying to confirm this both in vitro and from a few in vivo assays we have. And then to come to our glycosylation analysis, the glycans are certainly different between recombinant and virus-derived material. And I think this is something that we should consider when we look at recombinant material in the context of immunogens, especially. We see O-glycosylation at position 678. We didn't see any O-glycans or trace amounts at 2 other sites that had been variably reported among the different glycomics analysis papers. We didn't see any glycans there for what it's worth. And I think most importantly, we saw a significant amount of S1 shedding for the vaccine candidate expressed protein that we looked at. Again, that doesn't have a therapeutic site. excuse me, that does have the furin-completed site, that doesn't have stabilizing protein mutations. And as I said before, this is an important consideration for efficacy of structure-based vaccines. And I want to highlight the work of everyone. I think this is important, but I think there's been some amazing efforts. So I'd like to highlight everyone in the Zitzman group who participated in the glycan analysis project, in particular you and Snezhna, Anna, Fabian, and Manish, and Mario, who are involved in the mass spectrometry project, and of course Lucas and Matteo are helping us understand and interpret these data from a structural computational perspective. And thank you for your time.
That's great. Thank you very much, Weston. It was a really interesting talk, and really built on some of the themes we discussed earlier around the role that glycosylation plays. So I'm afraid in the interest of time, we are going to have to draw today's webinar to a close. So I'd just like to say thank you, everybody, very, very much for attending. Keep your eyes peeled for future Instruct webinars and events, and hopefully look forward to seeing you all either in person or virtually sometime soon. So thanks very much. Bye.